Assalamualaikum and hi. So today we are from Group 1. Will explain to you about Gaucher's disease. Okay, so this is our team members. We have Don, Edina, and me, Atira, Hanis, and also Nuri. Okay, first I will explain the nature of Gaucher's disease. Gaucher's disease is a lysosomal storage disorder that involves the sphingolipid degradation pathway. The core problem is a deficiency of the enzyme needed to break down glucose cerebroside. Because this lipid cannot be degraded, it starts to accumulate inside the macrophage. These swollen macrophages are known as Gaucher cells, as you can see them in the illustration here. Over time, the buildup of these abnormal cells causes enlargement and damage to several organs. The main organs affected include the liver, spleen, bone marrow, and bones. This accumulation disrupts normal organ function and leads to many of the clinical symptoms seen in patients. So overall, Gaucher's disease is essentially a problem of lipid buildup due to enzyme deficiency, leading to widespread tissue involvement.
The GBA gene gives instructions in making of enzyme called beta-glucosidase. This enzyme lives in lysosomes, which are responsible in breaking down the unwanted cells. Site of mutation is at 1q21, which simply tells us that the mutation occurs at chromosome 1 at the long arm, region 2, and band 1. Most common mutation type is missense mutation, where one DNA base change causes the whole one amino acid to change, leading to altered shape and function. Common mutation like N370S, which named as Gaucher disease type 1, so the non-neuronopathic means that the mutation does not affect the brain. As for L44P, which associated with Gaucher disease type 2 and type 3, neuronopathy means that it involves the brain.
Okay, so now we know that GBA gene mutation will eventually lead to the glucocerebrosidase enzyme defects. So one of the effects of the mutation is forming misfolded enzymes. As we all know it, that misfolded proteins are unstable and it is more prone to degradation by the proteasomes. Therefore, this will prevent the glucose cerebroside breakdown into glucose and ceramide. Next is the reduced catalytic efficiency. So gene mutation sometimes will allow the enzyme to reach the lysosomes, but it will reduce its ability to hydrolyze glucose cerebroside. This is mainly because of sometimes the active site will become distorted or the binding of the substrate has become weakened. So as a result, this will slow or incomplete the degradation, causing glucocerebrosidase accumulation. And lastly is the unfolded protein response. So the accumulation of glucocerebroside will trigger this cellular stress response. Um, so chronic UPR will eventually lead to cellular dysfunction and apoptosis and causes inflammation. So the inflammation will alert the macrophages to the inflammation site and releases its cytokines and also its systemic effects. So the macrophages will engulf or engorge these lipids, or we call it glucocerebrosidase, and it will become Gaucher cells. So it is called Gaucher cells because it has a special feature, which is wrinkled tissue paper cytoplasm due to the stored glycolipid fibrils. Okay.
Now, I will explain the protein coded by the mutated gene in Gaucher's disease. The protein is called lysosomal acid beta-glucosidase and is also commonly known as glucocerebrosidase. This enzyme is found in the lysosomes of macrophage. Macrophage are the cells responsible for breaking down waste material in our body, and lysosomes are like their digestive compartments. The main biochemical roles of this enzyme is to cleave glucocerebrosidase into ceramide and glucose. This breakdown step is essential because it maintains proper lipid balance in the cells. So, in simple terms, you can think of this enzyme as molecular groundskeeper. Its job is to prune or clean up excess lipid so that the cell stays healthy and organized. When this enzyme is deficient, as in Gaucher's disease, this lipid accumulate, leading to enlarged macrophage and tissue damage, which explain many of the clinical features of the disorders.
Now I'm going to summarize our presentation for today. The Gaucher's disease is caused by mutation in the GBA gene on chromosome 1q21, and these mutations commonly produces a misfolded or low activity form of beta-glucosidase. This enzyme normally breaks down glucose riboside inside lysosomes. When it fails, the lipid accumulate inside macrophages, leading to organ enlargement and the clinical features we see. For this presentation, we've gathered the gene level information from two reliable databases, which is Medline Plus Genetics and GeneCards. Medline Plus helped outline the disorder itself, its nature and clinical impact, while GeneCards provided a detailed view of GBA gene, the associated variants, and how these mutations affect beta-glucosidase. Together, these sources made it clearer how a single gene and a single enzyme can set off a cascade that ends in multiple organ involvement. That's all for our presentation. Thank you.